So this is the first talk on molecular biology. Today I will be discussing about polymerase chain reaction, PCR. PCR, it is actually the full-fledged or complete PCR reaction was done in 1984, and it is used to amplify a particular piece of DNA using or providing all the required elements including the enzyme and all the building blocks in a small tube, and providing the other conditions like temperature and pH, and this will amplify the piece of DNA which is supplied in the reaction mixture, and the target will be amplified to millions of copies within a few hours. And the use of PCR has increased nowadays in various fields like disease detection, paternity detection, and introducing mutations for the research purpose. So many uses are there. So PCR is one of the indispensable technologies in molecular biology nowadays. And the PCR and the conventional or the natural DNA synthesis in the cell, they differ slightly in the mode of action. In the case of PCR, we are providing an artificial atmosphere outside the cell. Everything is provided except a few enzymes which are normally used in the cells for the synthesis of DNA. For the synthesis of DNA in the cells, it requires a DNA polymerase enzyme, helicase is needed, primase is needed, ligase is needed. Different enzymes are used within a cell atmosphere. But in a PCR tube, we use only one enzyme, which is polymerase. Normally we use Taq DNA polymerase. Other conditions, the need of a helicase is removed by including a particular temperature step in the PCR reaction cycle. And also the PCR need external primers to be supplied. We will discuss those details in detail later. And the separation of strands, as I said, in the cell the helicase do this separation, whereas here we do this strand separation by heating. And also you know that in the cell when DNA synthesis happens, and one strand is synthesized continuously, the lagging strand is synthesized in pieces called Okasaki fragments. I will show you that in the next slide. And in the cell as well as in the PCR tube, DNA synthesis happens in the 5' to 3' direction. So it is towards the 3' direction the synthesis is happening, and it is replicating the other strand, that is a 3' to 5' strand, by adding complementary sequences. And also the DNA polymerase enzyme can start only from a double strand. So how that is acquired or happening in the cell as well as in the PCR tube, we will see later. And this is the picture showing the DNA synthesis within a cell. Here the DNA strands are separated by the enzyme helicase, whereas in the PCR tube this is done by increasing the temperature to 95 or 94 degrees Celsius. So the increasing temperature, all the bonds between the bases are broken. That way the strands are separated. Here helicase enzyme is used. And here in the leading strand, the synthesis is continuous from the beginning till the end, continuous DNA synthesis. Whereas the lagging strand, the synthesis in small pieces of 100 to 200 nucleotides length. And why? Because the DNA synthesis can happen only from 5' end to 3' end. So in the case of this one, the leading strand, this is a 5' end, so it can start from there. But in the case of lagging strand, the 5' end is here. So these strands are anti-parallel. So the 5' end is here. So the cell don't want to wait till the helicase completely separate the strand. So the synthesis should, in the lagging strand also, the synthesis should happen along with the leading strand. So the strategy is synthesize the length which is already separated. So it starts at the beginning somewhere here. That way this synthesized. As the strands are separated further down, again another site synthesis starts. again another synthesis starts. So this is it happens in pieces of 100 to 200 nucleotides length. And later these pieces are joined by ligase. But in the PCR2, there is no need of ligase enzyme because in both the strands, the leading strand as well as in the lagging strand, the synthesis starts from the beginning till the end. It's a continuous process because the strands are separated at the same time. The fully separated within a fraction of a second or within a few seconds. So the enzyme don't need to wait for the complete separation. So that is one difference between the DNA synthesis happening in the cell and DNA synthesis in PCR reaction. And how this PCR work? First, so we have a piece of DNA, double-stranded DNA. Then first we need to separate the strands. Here the separation is by increasing the temperature. You know that each and every chemical bond will be broken at a particular temperature. The reaction kinetics you should have learned about it. So when the temperature increases to 95 degrees Celsius, the whole double strand will get separated into two single strands. And from there, and as I said before, it needs for the DNA polymerase to start its action, it needs a double-stranded end. Here, what happens is we use primers. In the cell synthesized produce specific RNA molecules of short length by the enzyme primase. So here we supply the primers. So there are two primers for each PCR reaction. One for the leading strand, one for the lagging strand. Forward primer and reverse primer, which is complementary to the 5' end of the separator strand. That is, so the primer comes and attaches. That is annealing. So that is it will not happen at 95 degrees Celsius. 95 degrees Celsius, all the bonds are broken. So we reduce the temperature to a temperature which is suitable for that particular primer. For each and every primer, the annealing temperature varies because the sequence composition varies. The sequence, because like G and C, if there is more G and C, the annealing temperature will be slightly higher because there are more bonds between G and C, guanosine and cytosine, than A and T. So when the GC content is higher annealing temperature. So for each and every primer, we can calculate the annealing temperature. Now there are so many softwares available, free softwares are also available on the net. Using that, we can calculate the annealing temperature. And also, when you are ordering a primer, the supplier will also provide you an annealing temperature. Normally, they give a melting temperature. If they are giving 54 degrees Celsius as melting temperature for your primer, you use 1 or 2 degrees below that temperature as the annealing temperature. So you have to adjust, you have to standardize the annealing temperature for each and every PCR reaction, for each and every primer. So the primer has annealed here, and then the synthesis of rest of the strand. That is extension. So that also, there comes the enzyme DNA polymerase. Here we use Taq polymerase. Taq polymerase, the optimum temperature is between 75 to 80 degrees Celsius. But normally we use a temperature of around 70-72 degrees Celsius, or around 1 minute for every 1000 bases. So during when we increase the temperature again to 72 degrees Celsius, then the extension happens and the double-stranded DNA is produced towards the end of the extension process. Now the cycle restarts. So this is how the PCR reaction happens. So this is a typical or an example of a PCR cycle. First, there is an initial denaturation process. Here we give it a little bit longer period of denaturation or strand separation. That is 95 or 94 degrees Celsius. We heat around 3 minutes. This is for a normal Taq polymerase. But if we are using a hot start enzyme, this initial denaturation period will be slightly lower. It might be 5 minutes or 15 minutes. In certain enzymes, hot start enzymes, the initial denaturation should be 15 minutes. So that will be there. That is different for each and every enzyme we use. Then this is where the real cycle starts. That is a short period of 95 degrees Celsius where these strands break. So this first initial denaturation ensures that all these strands are separated. Because when we are using a DNA strand, it might be highly coiled in nature. So it might take a longer period of time for separation. That's why initially we are using a longer period of denaturation, 3 minutes to 15 minutes depending upon the enzyme. Then the cycle starts. In a typical cycle is around 30 seconds at 95 degrees Celsius. The strands are separated. Then you are decreasing the temperature to the annealing temperature of your primer. It can be 55 degree. It can vary between 55 to 65 degree depending upon the primer you are using or depending upon the GC content of your primer. So you choose that temperature and the temperature comes down to that level. And the annealing happens. That is a primer. The complementary primer attaches with the 5' end of the DNA strand. Then you are increasing the temperature to 72 degrees Celsius or 70 degrees Celsius, and depending upon the length of the pieces you are amplifying. If it is 1000 or less than that, you use around 1 minute. If the length is higher, we may use 2 minutes or higher, depending upon the length of the products you are amplifying. So towards the end of this process, all the DNAs are in double-stranded structure. So now you have produced the number of copies to double. If you started from 10, now you have 20 copies. Again, the denaturation, the temperature is increasing, 95 degrees Celsius, 30 seconds. So all these strands here at this point, all the DNA molecules are single-stranded now. Now the temperature comes down, annealing, primer attaches, temperature goes up, extension. So here, that is the end of second cycle. You started from 10 copies, then now you have, here it is 20 copies, here it is 40 copies. Like that, this is exponential increase in number depending upon the initial number of copies. Then, what are the components needed for a PCR reaction? These are the components you need in a PCR mixture. This DNA template is needed, of course, that is an essential thing. Then the primers for making the double-stranded ends. Then tag polymerase, the enzyme doing the job. Deoxynucleotide Triphosphates, that is the building blocks, the blocks by which the DNA molecules are produced. Then buffer, that is most important for each and every enzyme reaction, you know that. There is a particular optimum temperature. Here for this enzyme, this is around 8 pH 8 or slightly above that. That buffer will provide that. Then divalent cation. This enzyme needs the presence of divalent cation for its efficient functioning. Here we use magnesium or manganese ions. And monovalent cations to stabilize. It helps to stabilize the DNA molecule. Then, DNA template. What should be the nature of the DNA template? And it must be highly purified. Because if there is any chemical contaminant, it will affect your enzyme activity. You know that enzymes are very proteins, they get denatured easily. So the DNA should be very pure. So when you are isolating DNA, you use phenol or some other chemicals, so it should be free of that. And also it should not be containing RNA, it can affect your result. So salt, so if the salt concentration is high, it will affect the stability of the enzyme, so your reaction will be suffered. Then the concentration of the DNA template. The ideal condition or concentration is between 30 to 50 nanogram. Sometimes you may have to start PCR with a very low amount of DNA. Suppose you have only one cell, so then the concentration can go down. So there can be possible problems there, because when the copy is lesser, you have to use a higher number of cycles. So that when you increase a higher number of cycles, the sensitivity of the reaction might be affected. So 30 to 50 nanogram is the optimum concentration you needed. So you quantitate the DNA and take enough quantity. So higher concentration can produce false positive, because when the strands are when there is higher concentration, the number of strands will be higher. It can lead to a wrong primer attachment and produce false positive. And it is better to choose a piece of DNA with the GC content varying between 50 to 55%. And this is possible normally when you are studying an expression level of a gene. So you need to amplify only a part of the gene. But when you are going to clone a gene, you cannot have this choice because you have to amplify the whole gene. From the beginning till the end, you have to amplify. There is no choice of this GC. But if you are amplifying only a part of the DNA, you need to see this choice, 50 to 55% GC content part, which has this GC content, because this will help to keep the annealing temperature a bit high. When the annealing temperature is high, mispriming can be avoided. The primer will attach only to the specific site, only at the fully 100% complementary location. And this tag polymerase need to be activated. All DNA templates cannot activate tag polymerase. So the tag polymerase should recognize this DNA you supply as its substrate. That is the most important. So if the DNA is not recognized as the substrate by the tag polymerase, it will not be amplified. So the DNA template you choose should be treated by the tag polymerase as its substrate. So these are the important points regarding the template. It should be pure. The concentration will be 30 to 50 nanograms, and the GC content, the optimum GC content is between 50 to 55%, and it should be free from chemicals and other contaminants. Other contaminants means if you are purifying DNA, if you are using your bare hand, what can happen? The skins from your hand will fall into the DNA, the sample, and your DNA will also be there. So you may get a false result. So that is very important. You need to be very hygienic when preparing the DNA sample. And the tag polymerase should recognize your sample, your template as its substrate. Then how will you quantify the DNA? I said you should be using 30 to 50 nanograms of DNA. How can you quantify it? We normally use spectrophotometric method. Fluorometric method is also there. Spectrophotometry is quite easy. You use a UV light, you place the sample in the machine, and you measure the optical density at 260 and 280 nanometers. And you take the ratio of 260 by 280 ratio, and if the ratio is 1.8, that shows your DNA is pure, not contaminated by RNA or proteins. But if the ratio is low or higher, there is a chance there is a contamination by RNA or proteins. So you need to you quantitate this from the optical density. You can calculate what is the concentration of the DNA you have. From that you calculate and take 30 to 50 nanograms for your reaction mixture, and also it ensures the DNA is pure from the 260 to 280 ratio. And you know that when you are taking genomic DNA, only a small percentage of the DNA acts as target, and the rest it is non-target DNA. So you are concentrating on the targeted DNA. And the GC content, I have said 50 to 55%. Higher GC content increases the melting temperature. So it's very difficult to amplify if the GC content is too high, higher than 55%. And then, so now you have quantitated the DNA and then dissolving the DNA. You use stress power normally. You need to keep your DNA stabilized to have a good PCR reaction. And you should ensure complete dissolution. If the concentration is high, or if you are drying the DNA sample during the preparation too much, it is very difficult to dissolve. So, to ensure you dissolve, make sure that the DNA is completely dissolved. Sometimes you may use freezing and thawing a couple of times. That will increase the dissolution. So that is important, because if the DNA is not fully dissolved, what can happen? You might get a different reading when you are measuring, and you are not taking a different concentration. If the template number is too high, the dNTPs you provided will be acting as a limiting factor. So the concentration of DNA is quite important. So make sure that it is completely dissolved. And so I was talking about starting the PCR from a DNA template. Sometimes you may need to quantify the expression of certain genes in your cell of interest. So expression of genes start from the synthesis of mRNA, you know that, messenger RNA is produced. All those genes which are expressed will be converted into mRNA. That mRNA you are taking. If you want to use that mRNA in the PCR machine, you have to convert it into DNA. Before that, you have a separate reaction, reverse transcription. You are producing DNA from mRNA. You are using reverse transcriptase enzyme, providing the optimum conditions, and you convert the mRNA into complementary DNA. Short form we call it cDNA. So you can take either mRNA, pure mRNA, or you can take the whole RNA and do a reverse transcription reaction and convert all the mRNAs into DNA. So now you are getting a mixture of complementary DNAs. All the mRNAs are converted into DNA. From that, you amplify your gene of interest. For that job, that is to be specific for your gene of interest, you use specific primase. So you can also start from the primase. So as I said before, within the cell, as I said before, for the DNA polymerase to act, it can start only from a short stretch of double-stranded DNA. So within the cell, what happens is the strands get separated, and the primase enzyme produces a short stretch of RNA molecule that is complementary to the starting point of the replication. So that part that is all there. But in the PCR reaction, we are not using primase enzyme. Because of that, we have to provide a small stretch of DNA which is complementary to the 5' end of the strand you are amplifying. It's a short stretch of DNA which is complementary, and it is complementary to the 5' end because the synthesis is towards 3' end. And that is the primer. And you can design nowadays. There are so many softwares which are available free of cost online, like a Primer 3. Then NCBI site also you can have a software. And you can use any of those software and give your sequence, whether it is RNA or DNA, and the primer will be picked up by the software. And you can choose. Normally we choose a primer length between 18 to 30 bases. The shorter bases is good for higher amplification, but the primer is short. What happens is there is a high chance of mispriming. I will show that in the next slide. So designed using software, that primer is target specific, and you decide which DNA or which gene you want to amplify, and you give that to the software and say, okay, this is the length you want. Maybe you can choose. If you are using a real-time PCR, we use a short length, that is around 100 to 200 bases length. But if in a normal PCR, you can 300, 500 or 800, something like that, depending upon the gene and your interest. But if you are amplifying a gene, the whole gene for cloning, you have to amplify from the start to the end of the gene. So there is no choice for that. You just take the initial part of the DNA gene as the primer. Or if that part is not very good, what you do is you make a tail. That we will discuss when we are talking about primer production or primer designing. So it should be target specific, 15 to 30 bases length, because if the primer is short, high chance of annealing and efficient PCR product, but high chance of non-specific binding. So we use around 20 or more bases in a primer. Generally 10 picomole of each primer is sufficient for a reaction. 10 picomole is enough. But it is better to standardize the primer concentration, because if you are using excess primer, that can give wrong reading, wrong results. If you are using less amount of primer, it might be acting as a limiting factor, because if your PCR efficiency varies for each and every DNA piece. So for each piece you have to find out, standardize the amount of primer need to be used. You can use this as a model amount. You can either increase the concentration, but you can trial and error method. You can optimize the primer concentration. So this is how the primer pairing happens. So if you are using a very short piece of primer, what happens is, so in this case, you are using 3 bases. Never use 3 bases. Just as some simple model. So it has got a complementary place here and also here. So you will get 2 products. Then you slightly increase and still there is another site here. This here also 4 bases, again second site is there, 5 bases again, 6 bases again, a second place to attach for the same primer. So you get wrong product. Then increase, kept on increasing, but when you reach here, maybe 18 bases or 20 bases, there is only one complementary place. There is no other complementary places. So the length of the primer is important. But if the length is too high, like 30 or more than that, the annealing possibility, annealing percentage will decrease. So you use minimum number of bases, but you have to ensure optimum binding. Only single binding, no, not specific binding. That all these jobs will be done by the software. In the past, it was very difficult. You have to take this sequence before you would see each and every sequence and you design a primer. Now it is simple. You select the choice of your sequence, you give it to the supply to the software, and you provide the conditions like what percentage of EC concentration you want, what is the length of your product, you desired length, like that, and the primer software will pick that. All these will be satisfied. So DNA polymerase enzyme. This is quite important. So this catalyzes the elongation of DNA by adding nucleotide triphosphate one at a time to the 3' end. So it is the extension is towards the 3' end. So at the 3' end it is added. And when a nucleotide phosphate join with the existing strand, it will lose 2 of the phosphates. And DNA polymerase can only add nucleotides to the 3' end of the growing strand. That is why the direction is specific. And Taq polymerase. I think initial Taq polymerase was used in 1966. Yes, 1966. And it was done by Thomas D. Brock. He obtained this from a bacteria called Thermus aquaticus, from a hot spring at the Yellowstone National Park in the USA. So these bacteria are growing in the hot spring. The temperature is above 75 degrees Celsius. They can grow in that atmosphere. That means their enzymes are stable even at 75 degrees Celsius or slightly higher. But what will happen if you are using our enzyme in a PCR reaction? When you increase the temperature to 72 degrees Celsius, the enzyme will be denatured. So no reaction will take place. So this was the first hurdle faced by the scientists and the thermostable DNA polymerase that was obtained from Thermus aquaticus. But again another problem arose. Because when this DNA addition, the nucleotide addition happens, there can be a risk. By mistake, the enzyme can add another wrong base there. So it needs to be proofread and removed. So some of the polymerases, DNA polymerases, didn't have this proofreading activity, proofreading property. So we cannot use that enzyme. Because if you are using that, it will keep on adding errors. For each and every cycle, if you keep on adding errors, towards the end of 30 or 40 cycles, the number of errors will be too high. So you need an enzyme, DNA polymerase enzyme, which has got a proofreading activity. So a lot of searches, and finally we found this enzyme, Taq polymerase, as very suitable. And what the scientists did is, they isolate the polymer, the gene causing the production of DNA polymerase, the pol gene, and it was introduced in E. coli, which is very easy to grow in a lab. And this E. coli, because it is having that particular pol gene from the thermus aquaticus, it produces a polymerase which heat stability is quite high. And nowadays we are using that, and now we have lots of Taq polymerases supplied by different companies having different properties. Some enzymes are used for copying only a small stretch of DNA. Some are effective for even copying 10,000 bases. So depending upon your need, you can choose the enzyme you want. Some the fidelity is quite high, the error rate is too low, especially when you are cloning a gene. That is most important. The error rate should be very low. So hot start PCR, that is the latest trend. Not latest, a few decades before this was introduced. Because we set up a chemical, we are setting up a chemical reaction. That is, you are adding all the components needed for the DNA production. You have the template, you have the building blocks, the nucleotides, everything is in the tube. So the reaction can happen at a lower temperature also. Only thing is the speed will be slower. So that annealing at lower temperature can cause non-specific products. That is a problem in when you are using a normal enzyme, which is not hot start. You are setting up a reaction. Normally you use in ice, so the reaction will be very, very low. Still, there is a chance. But when we started using hot start PCR enzymes, this enzyme become active only when the temperature is increased for a certain period of time. So you have to heat it for 5 minutes. Certain enzyme you have to heat it for 15 minutes. Then only the enzyme will become active. So until that heating happens, there is no enzyme activity. There is no PCR product formation. So non-specific amplification is avoided. So this inhibits the activity of the Taq polymerase at room temperature. So you can set up the reaction even at the room temperature. You don't even need an ice bucket to set up this reaction. And this will enhance the data reproducibility. Each and every time you will be getting the same data. And in the case of hot start PCR, there are 3 distinct methods are applied to make this enzyme hot start. First one is you are using an antibody. So you are attaching this antibody to the enzyme, or the antibody is attached to the active site of the enzyme, so that the active site is blocked. When you heat this enzyme to a particular temperature, what happens? This antibody get denatured. These are protein molecules. They get denatured and they get separated from the active site, and the enzyme become active. Second is you are using an aptamer. Same principle. The aptamer is attaching to the active site of the enzyme until it is heated to a particular temperature. Then when the aptamer is removed and the enzyme site is free, and the enzyme become active. The aptamer is slightly better than antibody based hot start, because in the antibody based hot start you have to heat the enzyme for a certain period of time. Because when you heat this enzyme, even though it is stable, its activity decreases slowly. So you can avoid when you are using aptamer. You don't need to heat too much to remove this aptamer. A slight increase in temperature can remove this aptamer, and then the enzyme become active. Then you can also use specialized hot start dNTPs. You attach certain things to the dNTP, so that the dNTP cannot attach to the DNA template. So that is the third method. Normally we get hot start with this. So this is cheaper, and you can easily use in a normal laboratory condition. So using either antibody or an aptamer, and you get a hot start enzyme, and you set up the reaction, you heat the reaction for a certain period of time, then the enzyme become active and the reaction starts. So this is normal Taq polymerase. There is a second non-specific product is formed. But the same thing you did with a hot start polymerase, there is no non-specific product formation. So the proofreading activity, that is quite important, especially when you are amplifying a gene for cloning. Because if sometimes even a single base changes can destroy the gene, you may not be getting the correct product. So the bases are added, here what happens? The error happens. Instead of C, A is added. What happens is this enzyme has a 3 to 5 prime exonuclease activity. You need to see that the synthesis is from 5 prime to 3 prime edge. This exonuclease activity is 3 to 5 from here to that way. So the enzyme then read it, okay, and realize that okay, I did the wrong job. The wrong base is added here. It go back and cut that phosphate bond, and that nucleotide is removed. So this proofreading activity is quite important. Now you can have enzymes which will, the error rate is almost nil. Such enzymes are used when you are amplifying genes for cloning. The dNTPs. There are 4 types of dNTPs. Deoxynucleotide 5 triphosphate, DATP. Then GTP, guanosine triphosphate, thymine triphosphate, cytosine triphosphate. So all these will be provided at a particular molality. Adenine, guanine, thymine and cytosine. That is the nitrogenous bases. It has got a sugar molecule and 3 phosphates. When this is attached to the DNA, these 2 phosphates are removed. Then buffer. As I said before, pH should be maintained between 8 to 9. Normally we use pH 8. This stabilizes your DNA as well as provide the right optimum pH for your enzyme. And divalent cations are supplied. The enzyme activity is better when you give divalent cation. Normally we use magnesium ion or manganese ion. And Mn2 is better for PCR based mutagenesis, because when you are using magnesium, the error rate is lesser. When you are using manganese, the error rate can be higher and you can easily introduce mutation. And monovalent cations, we don't know the exact role of this cation, but they help to stabilize and enhance the activity of the enzyme. And complementary base pairing, you know that the bases are, the enzyme chooses or enzyme attaches the correct complementary bases, which is complementary to the leading strand or the lagging strand, depending upon where it is acting. This we have already discussed. Reaction mix. So how will you prepare a PCR reaction mix? So it needs your template, the dNTPs, that is the building blocks, the enzyme needed for this, the buffer to produce the optimum pH, and the cations. So normally we set up a 20 microliter reaction mix. We can even choose 10 microliter depending upon the cost of or your financial status. Or you can, if you need more DNA molecule to be amplified, more sample, more amplified products, you can set up 40 microliter. So starting for, if you are using a 20 microliter reaction mixture, it contains 1 microliter of Taq polymerase, and this is also varying depending upon the supplier. The essential thing is you need to provide 1 unit of the enzyme. Sometimes it may be in 2 microliters, sometimes it can be in half microliter. Depending upon the supplied product, you use 1 unit of the enzyme. Then 1 microliter of primase, 10 picomoles, 10 picomole of primer, 1 microliter of that. That also you vary depending upon your requirement and upon your primer. Then 2 microliter of 10x buffer. So this buffer is 10 times concentrated. Your mixture is 20 microliter, so you need 2 microliter. 2 into 10 is 20. So when it is in the full 20 microliter mixture, it is 1x. So 2 microliters of 10x. If it is 5x buffer, you use 4 microliters. If it is 20x, you use only 1 microliter. So normally we get 10x buffer along with the enzyme. The enzyme suppliers provide this buffer. And nowadays, this buffer comes with the divalent and monovalent cation. You don't need to add. In the past, we had to add this. Then dNTPs, 1 microliter of each dNTP in the picomolar concentration, 1 microliter of each of them. Nowadays you can buy a mixture, equal composition. So you add, if it is a mixture, you add 4 microliter. If you have separate them, you add 1 microliter each. Then 1 microliter DNA or cDNA. And your template, depending upon the concentration, you adjust the concentration so that you will get 30 to 50 nanogram. So the concentration, the volume may vary depending upon the concentration of your sample. And so now you have 1, 2, 4, 5, 6 microliter, and your total mixture is 20. Not 6. Here you have 2 microliter of primer, 1 microliter each. So 1 plus 2 is 3, 5, 6, 7 microliter. So to become 20, you have to add 30 microliters of DNA-free water. If you are adding water with the DNA, what will happen? It will chop your DNA, your template, and also your product will be chopped. You will not get any final product. So that is important. DNA, RNA free water or nuclease free water or DEPC treated water. You can make this water in your lab. And all this together, if your DNA volume is higher, you reduce the volume of water to a final volume of 20 microliter, and you take this in a thin PCR tube. That is quite important. The PCR tube should be very thin walled. Otherwise, the heat transmission will be very slow. It will affect the performance of your reaction. And so this happens. First denaturation, 2 strands are produced. So the primer annealing, then extension. So from 2 strands are 95 degree, 2 strands are produced. From there, so and the product length. So suppose in this reaction you are using a forward primer starting from here and a reverse primer using from here. So your product will be of this length. And if your primer is starting here, the product will be from this length. Or if your primer is starting here, it will be this length. So depending upon the primer you choose, the location, the complementary pairing of the primer, the product length will vary. So you have a product of from here to there. So the primer will decide the product length. So this is the whole process happening. Starting from one molecule, first cycle you have 2, the second cycle you will have 4, third cycle 8, 16. So likewise it increases, and finally within 30 or 40 cycles you get millions or billions of copies depending upon the starting number. Normally the cycle numbers you take between 30 to 40. The lower cycle number is better. As the number of cycles increases, what happens is the non-specific product formation is higher, the chance of non-specific product formation is higher, and also if in one sample you have more DNA and one sample you have lesser DNA, what happens if the cycle number is higher, the sample with the DNA, higher amount of DNA, the dNTP may act as a limiting factor. So their amplification will be slowed down. So you are getting a wrong result. So the minimum copy cycle number is better. But if your copy numbers are too low, you have to use higher cycle numbers. Depending upon your copy numbers, you have to standardize the cycle numbers. First start from the lowest number of cycles. If you are getting enough amplification by the lowest number of cycles, that will be better. So in a PCR, providing the optimum conditions, and if you are careful about the purity of the DNA, you can amplify the product without any problem. Within 2 or 3 hours, you can getting millions or billions of copies. Either you can decide the expression level, or you can use this amplified gene for a cloning process. For a cloning process, what you need is you need to purify this. You have to remove all the unwanted components except your desired products. Or for the gene expression studies, you have to see how much is produced. For that, you will run a